So continuing here with the electron transport system, again, we typically have to remove hydrogens from organic molecules. They get then slapped onto NADH, FADH2, and get split into electrons that will be passed down in these electron transport chains. And then the hydrogens typically are being used in a mechanism of chemiosmosis. In the end, we're going to have to put it all back together. These electrons, they have released all their energy, and so they need to find a final electron acceptor, and that is typically oxygen. So we then take oxygen. So this will be oxidative phosphorylation. We need oxygen as the final electron acceptor here. And then these hydrogen ions, they will be also added on to here so that we make what's called metabolic water. And this process releases quite a bit of energy. So we're making quite a bit of ATP, which is what we wanted. A spin-off of all of this is photophosphorylation. That's essentially photosynthesis, where you are using also electron transport chains to make a lot of ATP, but the initial energy source to lift these electrons into a higher energy state is because of light. And so what you would see here would be in photophosphorylation is that light will excite electrons off of chlorophyll, for example, as a pigment molecule. And then these excited electrons, they go down these electron transport systems and also generate ATP as well. So these are basically all spins off of the same thing, using somehow getting electrons that have a lot of energy and then using them in these electron transport chains to make ATP. And we'll get more specific examples later. Okay, so metabolic pathways of energy production. They involve these whole enzymatically catalyzed pathways that ultimately lead to the production of ATP. They typically take energy from organic compounds by removing the hydrogens and then running these electron transport chains to convert the energy in form of ATP, cellular energy. Okay, so here, these are our coenzymes that are necessary. A lot of times we need to take these hydrogens off of organic molecules, and we need to somehow get them to the electron transport chains where we can use them then for the energy conversion process. And so these hydrogens, they get removed from organic molecules and then slapped onto NAD to make NADH, this one right here. And then the NADH, this molecule, will then move the hydrogens to the electron transport chain, where we then rip them apart into hydrogen ions and then the electrons that we need to make all that ATP. So anyway, the goal will be to add these inorganic, or to add phosphates to ATP one way or the other, and generate a lot of ATP. The most efficient one of these processes to make ATP, it's oxidative phosphorylation, because there the end products will be very low energy CO2 and water, but we must have oxygen typically. There are some alternatives, and bacteria sometimes they can use sulfur as well, but for the most part we need oxygen as the final electron acceptor to receive those now low energy electrons and the hydrogen ions, and put all of this back together to make water, metabolic water. And then, of course, these organic compounds that have lost all of their hydrogens, well, then what's left is the carbon and the oxygen, and that would make CO2, which is very low energy. Okay, so let's move on. We're ready now for carbohydrate catabolism. That means we're using carbohydrates that have quite a bit of energy, and we are breaking them down to CO2 and water, ideally, in the most efficient pathways to generate ATP. Now, if we do have oxygen available and we can go all the way, then we can use a three-part process that's known as oxidative metabolism. And it starts with glycolysis as our part one, moves on to the Krebs cycle as number two, And then finally the electron transport chain that then churns out lots and lots of ATP. Now, again, this will require the presence of oxygen. So if there's no oxygen available, or we have an anaerobic metabolism, organism, then we are limited to glycolysis, which is basically results then in byproducts, and this would be fermentation. But if oxygen is available, then we can run the Krebs cycle on the electron transport chain, which is much more efficient. So here, a diagram that kind of shows the different pathways. If there's no oxygen available, or we have an organism that's anaerobic, then we are limited to fermentation, which is a partial breakdown of a carbohydrate into pyruvic acid. So we can use glycolysis, and then we end up with some sort of byproduct of the fermentation, some sort of end product. So we need these fermentation end products, and that's typically either alcohol, so ethyl alcohol, or it could be lactic acid, that's a popular one, or some other small organic acid typically. Now, over here, if we do aerobic respiration, then we're going to go from glycolysis. We still produce the pyruvic acid, but instead of sort of going into this final product formation right here, we are going all the way oxidizing these food molecules, these carbohydrates. And so we're going to use the pyruvic acid, convert it into acetyl-CoA, which enters the Krebs cycle. There we're completely oxidizing the carbohydrate in this case, removing all hydrogens that were on that organic molecule, slapping them onto our coenzymes NADH and FADH2. And then these electron carriers right here, they will move the hydrogens to electron transport chains, where we then split the hydrogens into electrons and hydrogen ions. In the end, the electrons will release all of their energy. We need to find a final electron acceptor, which is typically oxygen, and then make metabolic water and lots and lots and lots of ATP. So this is much more efficient in terms of breaking down these food molecules completely to CO2 and water and gaining a lot more ATP, but not every organism can do it. Oxygen is not always available, and so sometimes we really don't want full oxidation, so we're happy to generate some alcohol, for example. So if you want to make some good wine, or also bread and cheese involves fermentation pathways, where you then actually appreciate the final products of the fermentation pathways. Now let's take a closer look at glycolysis. Basically, it is the conversion of glucose as a starting compound to pyruvic acid. That's the end product. It's not in one step. There's a whole set of enzymatic reactions that need to happen. But in essence, it's the process of going from glucose to pyruvic acid. It produces actually a grand total of 2 ATP. That may not sound like a lot, and it really isn't, but it's better than nothing. So in order to go from glucose to pyruvic acid, you have to first put in 2 ATP. Then you get out a grand total of 4 ATP, but you have to subtract what you had to put in. So minus 2 ATP, so you end up with 2 ATP net gain out of glycolysis. But let's take a look at this. So here we have the 2 ATP input that I just mentioned, and then we are going to get us an end product that's pyruvic acid. Here's a little bit more detail on the glycolysis pathways. So we're starting out with glucose, and we're ending up with pyruvic acid, just to keep things in perspective. But you can see there are quite a few steps involved. You don't need to know the intermediate right here, but you should know. So we need to first put in 2 ATP. So basically we're slapping a phosphate onto the sugar molecule, which has 6 carbons, if you can see that right here. So one of the phosphates, so here we're converting an ATP into ADP, and that third phosphate was actually added onto here, so one end of the sugar molecule. And then we're going to take another ATP and slap on a phosphate on the other end of the sugar molecule, so, and I call that it's kind of supercharged glucose at that point. And now we basically break the glucose in half, and then you're going to have 2 3-carbon compounds, and you go through another set of conversions right here. You end up getting 2 ATP out here and another 2 ATP out here, but don't forget that you had to put in 2 ATP, so your net gain is 2 ATP, and you have converted the glucose into pyruvic acid. That's basically what you need to know about the glycolysis reaction. So starting compound glucose, end product here of glycolysis, pyruvic acid, and then the energy, from an energy balance sheet perspective, you had to put in 2 ATP right here. You got 2 plus another 2 out here toward the end. So your net gain from glycolysis is a total of 2 ATP. And so let's take a look at this down here. So glycolysis, we are starting out with high energy carbohydrates such as glucose. We're ending up with 2 pyruvic acid molecules. We had to put in 2 ATP, but we got 4 ATP out. So our net gain is 2 ATP, but overall 4 were of course produced. And we also gained 2 NADH from a partial oxidation of the original glucose. So here, the glucose, basically in glycolysis, we're starting out with glucose, we're ending up with the 2 pyruvic acid, so the 2 actually should be right here, 2 pyruvic acid. So we had to put in 2 ATP, we got 4 ATP out. That means we have a net gain of 2 ATP per molecule of glucose that went into glycolysis. Now, here are some side tracks, sort of alternative spins off of the traditional glycolysis that we just talked about. There's also a pentose phosphate pathway. That means we're not using a 6-carbon glucose type of sugar, glucose or fructose, but we're using a pentose sugar that has 5 carbons, and that one produces also NADPH, and it can run simultaneously with glycolysis. And then there's something called the Entner-Duderov pathway. That one produces NADPH and ATP, does not involve glycolysis, so it's an alternative pathway that occurs in Pseudomonas, Rhizobium, and Agrobacterium. Now, ideally, we should be able to oxidize that pyruvic acid further, because there's still a lot of energy in the pyruvic acid, and so if we have oxygen available as the final electron acceptor, then we can run the Krebs cycle and the electron transport chain, gain a lot more ATP, and have basically completely oxidize our original glucose. So in cellular respiration, now we are liberating more electrons, and we can operate what's called the electron transport chain. The final electron acceptor in most cases is oxygen, but microbes, they have some genius other ways, some alternatives to that. The human body can only use oxygen as a final electron acceptor, but anyway, we're going to take a look at that, and we're generating lots and lots of ATP. So basically, you should never be happy with just glycolysis. It just doesn't generate enough ATP, and also it leads to byproducts, because the pyruvic acid has to be converted to something, and it's typically the alcohol that could accumulate and end up being a poison, or it could be some other organic acid like lactic acid that you're going to feel in your muscles if you are not feeding your muscles enough oxygen. So ideally you want to move on in cellular respiration, and so ideally here aerobic respiration, the next step then will be the Krebs cycle. And there we take the pyruvic acid that we generated in glycolysis, and we need to convert it into something called acetyl-CoA, which that can then enter the Krebs cycle. So the preparatory step here before we can enter the Krebs cycle is we need to take the pyruvic acid, we need to oxidize it a little bit, so we're losing 1 CO2, and then we are down to a 2-carbon compound that then gets converted to acetyl-CoA. We also get 1 NADH out of the whole thing, and then we're ready to go into the Krebs cycle.